Good morning, everybody. I hope you've all found your seats. I see that you're sort of clustered regularly and well interdispersed. I'll keep this very short. I'll keep the palindromic repeats and puns to a minimum. We are here today joined by the wonderful Jennifer Doudna, who we're very honored to host here for the second annual installment of the Sjöstrand lecture series on structural biology. So without much further ado, please give Jennifer Doudna a welcome, CRISPR, fantastic applause.
Okay, we're right on the nose, 9:30, so I'll time myself. I don't want to talk for too long. First of all, thank you very much for that introduction. It's a pleasure to be here in Stockholm. And I hope you can all hear me. Can you hear me in the back? Okay, yes? Good. So I was asked by Alexei to speak a little bit about my scientific background. And so what I thought I would do is just tell you a little bit about where I grew up, what I thought was interesting when I was growing up, and then how I got interested in the particular type of science that I'm doing. And certainly I'll start by saying if you had asked me almost at any point along my career path what I would be doing even a few years ahead of that point in time, I would have had absolutely no idea. So, my path has been circuitous, shall we say, but I've always been driven by just really enjoying doing science and knowing that I loved the process of discovery. So I've always just kind of tried to pursue that, and I've tried to do it with people that I found compelling. And I've been incredibly fortunate that I've just, had great mentors along the way, and I've also had amazing people that have worked with me in the lab up until right now. So that's all been, really a fantastic experience. So I grew up in Hawaii. I grew up in the state of Hawaii, the 50th state in the US. It's out in the middle of the Pacific Ocean. My dad, we moved there when I was 7. My dad was a professor at the University of Hawaii. It's a state university. And he wasn't even at the main branch of the university. He was on an outer island, we called it, island of Hawaii. And he was a professor of American literature. So not a scientist. And so we moved there when I was 7, and I grew up in a very interesting culture. It felt kind of foreign to me. So we had moved from Michigan, so it was a big cultural change. And I certainly felt the kind of the isolation of being out in the middle of the Pacific Ocean. I often had this feeling when I was growing up that life was going on everywhere else, but not where I was, right? There were all these things happening in the world, but they weren't happening around me. So I often had this feeling that, that I was sort of in a really a backwater kind of place, which I was. But it was a beautiful backwater place. And I was very much influenced by the natural world in Hawaii. So it's an interesting natural environment because a lot of organisms, plants and animals that have found themselves in Hawaii either by they've got there on their own or they were brought in, introduced by humans, they end up evolving in this relatively sequestered place. And so they have interesting properties that give them advantages in that island environment. And I didn't know anything about DNA at that time or think anything about it, but I certainly noticed that there were a lot of interesting plants and animals that you didn't find other places. And then I wondered about that. Why is that? How does that work? What gives them unique properties? And how do they evolve in these special ways? So I just found myself wondering about that as a kid. And then, let's see, I think I was about 12 years old when my dad, a literature professor, so he loved reading. He read sort of everything. He loved reading about science, even though he wasn't a scientist. And he would often, poke around in used bookstores and buy old books. And he bought a kind of a dog-eared copy of Jim Watson's The Double Helix. And he threw this book on my bed, and I came home from school one day, and there it was. And he said, I think this might be an interesting book. And it took me a while. I didn't pick it up for a while. I didn't, double helix, what's that? I didn't know what that was. And I didn't know if it was interesting or not. But I don't know, some rainy afternoon, I thought, okay, maybe I'll read this book. And it doesn't take very long to read it, as you know, if you've read it. And it kind of blew my mind because it was the story of the discovery of the structure of DNA. And not only that, it was a very human discussion. As you know, if you read the book, and it was a bit controversial at the time. I only found that out later, but it was really a discussion about not only the science of what was done, but also the personalities involved and the people. And I found that to be fascinating. And I started imagining, I started thinking, gosh, it's amazing that you can do experiments to figure out the structure of a molecule. I had no idea that that could be done. And at the same time, I was getting into high school, and then a couple years later, I took a chemistry class in high school from a wonderful teacher, Miss Wong. I don't know her first name, but Miss Wong was how I knew her. And she taught us kids in this chemistry class that science was not about memorizing a bunch of facts in a book and then regurgitating it on a test, but it was actually about the process of discovery. And it was about figuring things out. And I loved solving puzzles. I mean, that was something we kind of did a lot around my home. My dad, again, loved puzzles of different types, and we were often doing jigsaw puzzles at home and crypto quotes and things like that. And so I kind of started to put two and two together, and I also knew I loved math, and I really enjoyed thinking about the chemistry of life, I guess. And I started to have this fantasy about what if I could do science in the future. And frankly, I didn't know any scientists, and I certainly didn't know any women scientists. I'd heard of people like Rosalind Franklin and Marie Curie, but I didn't know anybody in my world sphere that was a scientist. But it seemed like a really interesting thing to do. And so I started to imagine how I would do it if I wanted to do it. And I thought, well, I guess I'll go to college and I'll major in chemistry, but I knew I was really interested in biochemistry, kind of the application of chemistry in biology. And at that time, this was in the late 1970s, early 1980s. It was kind of a rare thing, but fortunately, I found a liberal arts college on the West Coast called Pomona College. It's one of the Claremont colleges. And they actually they had started, I think maybe the year before I applied, a program in biochemistry for undergraduate students. So I applied there, I got accepted, and I went, and off I went. And I went sight unseen. So in those days, I don't know how it is here in Sweden, but nowadays in the United States, and I have a son who's looking at colleges, the parents will take their kids around, touring, looking at colleges and campuses to try to think about where they might want to go. None of that happened, at least for me. My parents didn't have much money. We were out in the middle of the Pacific. They didn't have money to travel, so everything, and we had no internet in those days, right? It wasn't like I could Google. No, it was all just, writing away and saying, please send me your application package, filling it out, and then getting a letter later and saying you're accepted, and then getting on a plane one day and flying there and thinking, I hope I like this place because that's where I'm going. And while I was there at Pomona, I struggled a bit, to be honest. The first year was really hard, and I missed home a lot, and chemistry class was really tough. I was in with a lot of students that were very smart and very accomplished, and many of them had gotten 5s on the AP chemistry test. Again, I don't know if you do that here in Sweden, but in the U.S. the students all take these advanced placement classes. Well, my public high school in Hawaii didn't have that. Right, so I had not done any of that. So, I found myself really, swimming upstream, so to speak. But the second year of college, I took organic chemistry, and I absolutely loved it. I loved it. It was, it was the process of solving problems. It was, how do you make this compound? We're going to give you a few starting materials, but you have to get to this complex molecule. Loved it. It was, fascinating and really fun. And I also loved the lab, working in the lab. I found that to be also really cool. You could, mix things together, and if you did it just right, and sometimes you could even get things to crystallize, and, you could make these compounds, and then there were all sorts of ways to physically analyze the compounds. And it was, it wasn't biology, but it was really interesting. I loved doing it. So I felt like, okay, I'm on the right track because this is stuff I really enjoy doing. And then in my junior year of college, I got fortunate that my biochemistry professor announced one day that she had 2 spots in her research lab over the coming summer for undergraduate students to work. And I had no idea what she worked on, and I hadn't ever worked in a lab before except in my coursework, but that sounded amazing. And so I applied to do it, thinking, well, I'll never get it because I'm sure everybody here wants to do it, but I'll throw my hat in the ring. And lo and behold, I got one of the spots. And so I started, working in her research lab, and it was just an incredibly wonderful experience. Working over the summer, about 10 weeks, working on a project. We were actually working on Myxococcus xanthus, if you've heard of that. It's a bacterium that grows in the soil. And it's very interesting because it undergoes a developmental pathway. So when it finds itself in nutrient-limited conditions, it forms these fruiting bodies that you can actually see by eye, but you can also see them under the microscope. It's a little bit like dictyostelium, if you've worked with that. And so the question that she wanted to ask was, what's the chemistry of that process? How do the cells tell each other, hey guys, we're starving, let's form these spores or fruiting bodies. And so how do the cells know to swarm together to form these structures? And so it was a process of trying to do classical biochemistry, of isolating molecules from these bacteria and trying to ask what's different when they're growing in nutrient-rich conditions versus what happens when they're starving. And the very first experiment, the very first thing that had to be done that summer was, we need a way to grow a lot of these cells and get them to form fruiting bodies so that we can start purifying molecules from the cells. And so she had hired a technician a few months earlier, and this guy was an older, he was out of college already, and he'd been working in the lab for a few months. To me, he seemed, wow, very experienced. And so my professor said, well, we need to get this working, but so far this technician has been trying for the last few months to get these cells to grow on these big sort of baking pans with agar on them, sort of minimal agar, and he hasn't had success. And so I'd like you to try this. And I thought, oh, great. It's something that this experienced person's been doing. It hasn't been working. And now there's little old me. So, I did some reading and I put together a protocol. And she was, of course, helping. My professor was helping me do this. And, we poured the agar into these baking pans and spread the cells and covered it with foil and, let the cells grow overnight. And I still remember, waking up the next morning in my dorm room and thinking, oh, I can't wait to get to the lab to find out, what's happening. And I get to the lab and I peel back the foil on this big baking pan and I see these little structures, And you could tell, I didn't know what a fruiting body was, but you could sort of tell that this looks like some kind of a structure. And so I showed this to my professor and she said, oh my gosh, this worked. This is what we want. We wanted to be able to grow these cells. And so that was my first kind of, experience with sort of success. And I didn't know what I had done right, but probably it's kind of beginner's luck. But from there on out, it was just this process of doing things. And of course, not every experiment worked. In fact, many experiments didn't work. But to have that sort of feeling of I can do this was very motivating for me, very motivating. And then sort of fast forwarding, so then I went to, after college, I was always thinking about, I knew I loved doing science. How can I continue to do more science? And I came from an academic family, so, my family was always about, getting more education. So I thought, well, maybe I'll get a PhD. And so, okay, I'm going to get a PhD. Where do I apply? I didn't really know. And so I applied to a few places, and I ended up getting accepted to, I applied to my dad's alma mater, which was the University of Michigan, state school, and a couple others like that. And then I also applied to Harvard Medical School. Why did I do that? Well, I had a lot of my classmates in college were pre-med, and a lot of them wanted to go to medical school. And sort of the pinnacle for them was Harvard. Harvard Medical School was like everybody wanted to go there, and almost nobody ever got in. But I was applying to the graduate program at Harvard Medical School. So it was a lot easier to get into the graduate program at Harvard Medical School. And, so I got in, and, I I asked my professor, what should I do? And she said, well, I think if you go to a place like that, you're just, you're going to be connected. You're going to immediately get connected to so many people because Boston is such a center for research and everything. So being this, girl from the backwater state of Hawaii in this little tiny town, that was kind of appealing to me. I thought, wouldn't it be cool to be kind of feel like I'm in the center of things for once? So I went. And then, I got into, I did some rotations as we do, and I got into a wonderful lab, the lab of Jack Szostak, who was a person 12 years older than me, right? So he was, I was 21 or something when I joined his lab. He was 33, already a tenured professor at Harvard, right? So, and he was widely kind of acknowledged at that time as a kind of a genius, and the thing about him was that he was very hands-off in the lab in a way. So he gave a lot of us in the lab freedom to do what we wanted to do, but he was very passionate about his work. So, if you ever had an idea or a result and you wanted to share it with him, he was always kind of there, and he would get very excited about things and say, well, oh gosh, that result, that probably means that we could then do this or that, And the problem that he was working on was about the biggest, craziest, hardest problem you could imagine. It was the question of, the origin of life. Like, can we figure out the chemistry that led to the evolution of life on Earth? And I couldn't think of a bigger or more interesting question in a way. But it was also, how do you break that down into experiments that you could actually do, that a student could do and eventually get a PhD on? It sort of seemed like a, incongruous thing. And yet this was his genius, or one of them. One aspect of his genius was that he did that, right? So he posed this really big, really, really interesting question, but then he broke it down into experiments that we could actually do in the lab. And so I started working on ribozymes, so RNA molecules that had been discovered not too long before that by people like Tom Sheck and Sidney Altman, who was there who was at Yale at the time, to figure out, whether you could learn something about self-replication and self-replicating systems by studying these catalytic RNA molecules that didn't catalyze synthesis of RNA, and yet you could sort of imagine a way that they could be rejiggered to do that. And maybe you could imagine even a path by which something like that arose on the early Earth. And that was the kind of the topic that we were studying. And so, I got going on this, and I still remember one day when I was sitting at my desk in the lab, and I think I was I was probably a first or second year grad student, and struggling with one's experiments as one does. And my advisor came over to me and he said, "Hey Jennifer, I've been thinking about your project, and I was thinking about an experiment that might be interesting to do, but I'm not sure, and I wanted to get your opinion on this." And I was stunned. I mean, here's this guy who's a kind of a genius and tenured professor at Harvard. He's asking me, some lowly grad student who's, just starting out and struggling with my experiments, what I think of his idea. And I realized later, I don't know if he did it on purpose or not, but that was an incredibly important moment for me because it was so validating, right? It made me feel like I mattered, like he cared about my opinion, and he really wanted to know what I thought about it. And we got into a discussion and we debated, and and he was very open to me saying, well, I think maybe we should try this experiment this other way. And he would say, well, why? And we'd debate back and forth. And he wasn't shy about criticizing me too, which was good. But we had plenty of debates back and forth where I felt like I was kind of intellectually validated and valid. My ideas were mattered. And that was so, I've never forgotten that moment. It was so important for me. And I think that's something that I would say to all of you is to, as you go forward, to first of all, look for people that give you that kind of validation, because I think it's really helpful, especially when you're starting out. And secondly, as you go forward and you start to become a mentor yourself for younger people who are coming along, look for ways to do that with them as well, to help them feel validated, like their views matter. And by the way, I read I was reading an article at some point about Google, the company Google. And lots of articles written about how does Google, a company like that, how do they kind of encourage innovation? And they had all these teams at Google that were put together with people in the company to address problems that they were trying to solve at the company. And as you can imagine, some teams turned out to be consistently more effective than others. And so the question was why? And so the company didn't know why. And so they hired a psychologist to come in and spend 2 years at the company looking into this. And she wrote an article about her findings. And I thought her results were extremely interesting. It turned out that it didn't correlate with things like, having the most experienced people on the team or having more of a certain type of person on the team. What mattered was the culture of the team. If the culture of the team developed so that every member of the team, no matter how experienced or inexperienced they were, felt enabled to contribute, that team was more successful over time than others. Isn't that interesting? I think that's fascinating. And so that means that it really matters to have contributions from people from diverse backgrounds, experienced, inexperienced. I often find in my own research lab that some of the best ideas at our group meetings come up from the lowly undergrad, sitting in the back of the room, some freshman, who kind of shyly raises their hand and says, well, why are you doing this experiment? And everybody's suddenly like, yeah, why are we doing this experiment, right? Great question. So I think that's something that I've learned over the years, is that it's not only was incredibly important for me personally to feel validated, but it's important as mentors to do that as well, to make sure that people feel enabled to contribute to things. Let's see how I'm doing on time. Okay, so it's 9:51, so I'm just gonna maybe just say a few more things. Let's see. So, I was in grad school, working on ribozymes and catalytic RNA. And then, I got to towards the end of my graduate training, and I felt, on the one hand, the work had gone really well. And, we had been able to do some really interesting things with ribozymes, with self-splicing intron RNAs, getting them kind of rejiggering their chemical activity so that they would actually ligate together little pieces of RNA on an RNA template. It starts to sound like a replicase, right? Kind of a polymerase. And that was really cool, really interesting. And you could start to imagine how an activity like that could have emerged at some point in the evolution of life on the planet and could have contributed to kind of the RNA world hypothesis, the idea that RNA molecules are potentially capable of serving as templates, but also being the catalysts of replication. But there was something very frustrating about my work in grad school as well, and that was that we had no idea what we were working with. Nobody had ever seen an RNA molecule except for tRNA, and that work had been done in the 1970s. This was already the late 1980s. And so I was thinking to myself that, if I really want to understand this system, we need to know what these molecules look like. And there was kind of increasing evidence. People debated at the time. It sounds shocking, especially, for those of you that have worked on structures of the ribosome and things like that. But at the time, people debated, would RNA really have a defined three-dimensional structure? And some people who were very knowledgeable and very experienced said, absolutely not. I mean, this is going to be a floppy polymer. There's no evidence that it would form a 3D structure. Other people said yes. Tom Cech was one of them. So his lab had generated some biochemical data that suggested that these self-splicing introns, in fact, had 3D architectures, but, nobody knew. So I started to have this crazy idea. What if you could crystallize RNA, just like people have crystallized proteins? And there was this one example of crystallizing tRNA, and again, many people said, well, tRNA is kind of special, so probably most RNA won't behave like that. But who knew, right? And so I sort of I started thinking that this would be something that would be really interesting to try to do. And believe me, there were a lot of people that said, well, that's the end of your career. That's it for you. You're going to go off and try to do this and we'll never hear from you again. And, it was scary. And I thought they might be right. but I'll never know if I don't try. And I think, partly because of, coming from the kind of background that I did, I mean, nobody expected me to do anything. Like, my parents didn't expect me to have a career or, be a certain kind of person or be a scientist or anything like that. So at some level, I felt like I kind of have nothing to lose because no one's expecting anything of me. So if I try something crazy and it doesn't pan out, I'll figure something out. I'll do something else. I'll figure something out. I'll do something else with my life. I'll try a different kind of science. So I decided I would try to do this, try to crystallize RNA and crystallize, RNAs that I was working on, these sort of self-splicing intron-related RNAs. So where was I going to do that? And, I talked to some of the top crystallographers. Tom Stites was somebody who I talked to at the time about, could I come to his lab and do this? And he was certainly interested. But I decided in the end that it would be best if I I should pick the very best RNA biochemistry lab that I could think of, because it was probably really a biochemistry problem. If you could purify these molecules and, figure out how to crystallize them, then getting the actual structure would be, something that you could figure out technically. Now, little did I know how difficult that would be, but anyway, but that was my thought process. And so for that reason, among others, I decided to apply to Tom Czech's lab in Boulder, Colorado. And fortunately, he accepted me, and I ended up going there for my postdoc. Boulder is a fabulous place. If you've ever been there, it's a wonderful, beautiful place. First thing I did was buy a mountain bike when I got there, and a pair of cross-country skis. I could ski out my back door in those days in the wintertime, and bike to work every day. It was great. And I started working on this challenge of crystallizing RNA. And fast forwarding, that basically, we had success, right? I mean, that ended up working, and again, I had to deal with. There were lots of challenges there, lots of things that didn't work. Believe me, many, many times, and I would go for weeks at times thinking, this is a bust. This is never going to work, and everyone was right. This is a disaster, and but I still kept going because I am very stubborn. And I desperately wanted this. I desperately wanted to see these RNAs. I really, really did. And in the end, we were successful. And part of it was that Tom Stites, after he had offered me a postdoc, I had turned him down and gone to the Czech lab. He ended up coming to Boulder on sabbatical with Joan Stites. So they came for a year on sabbatical when I was a second-year postdoc. And I had just gotten these crystals, but they were very difficult to work with. They didn't behave very well. They decayed very rapidly in the X-ray beam. And it wasn't clear that one could ever do anything with these. And at the time, so Tom, if you know Tom and Joan Stites at all, this will resonate with you. So Joan, being the very diligent, conscientious person that she is, spent almost that entire sabbatical sitting in her office, typing papers and typing reviews, reviewing people's theses, all that kind of stuff, right? So we didn't see much of Joan. Tom was always in our lunchroom with a cup of coffee, right? He was sort of there ubiquitously. So I could always find Tom Stites around. And I told him about these crystals. And he said, well, there's this kind of new method of cryocooling that's going on that was kind of developed originally by Otto Jonath for dealing with very difficult crystals of the ribosome. But he said, in my lab at Yale, we're starting to get this working, and maybe this would be something to try with these very fragile ribozyme crystals. So he arranged for me and a technician from Boulder to go to New Haven, to Yale, to try this. And we worked with David Gerasolmi, who was a student in his lab at the time. And bottom line, it was a fabulous success. So we were able to, again, very technically tricky at the time to do this. He had a little plunger, it was like a dipper. You had to plunge your crystal into liquid nitrogen quickly and then pry it out with tweezers and get it on. So there were a bunch of technically tricky things to do. But in the end, we put this cryocooled crystal on the X-ray beam. And this was just a rotating anode, right? No fancy, no synchrotron, right? This was just a R-axis rotating anode. And we got 3 angstrom diffraction. And it was just, it was one of those moments in your life when, you've worked for, at that point, several years had kind of built up to this moment for me. And seeing that, I still, I feel it right now, just this sense of, oh my God, this is so incredible. This is so amazing. And, it was just that kind of a moment for me. And I've only had a few in my career, but that was one of them. And that set me on a path for the next bunch of years, because then, we had to figure out the phase problem, so we worked on that, figured that out. That was done with Jamie Kate, who came to the lab, my lab at Yale, when I had started my own faculty position there. Figured that out using osmium hexamine. Turned out to be a great compound for phasing these crystals, and that ended up working out as a way to phase the ribosome crystals later as well. And because we were one floor below Tom Stites and Ned Bond, who was there at the time, and Paul Nissen, were able to walk downstairs and get osmium hexamine from us and do the initial soaks, and that also worked for phasing the ribosome. So, there was just a bunch of really interesting, great science that came out of all of that through our work and collaborating efforts as well. And, and then you'll hear today, later, if you come to the seminar today, a bit about our work on CRISPR. But, the only thing I'll say there is just that, again, if you had asked me at that time, certainly, if I could have imagined where our work would have gone, I would have had no idea. And even a couple of years before we did the work with Emmanuel Charpentier, I would have had absolutely no idea where that would have gone. So, we've always just been driven by a passion for discovery, for doing work that we found interesting, curiosity-driven science, question-driven science, it's been fun. So I'll stop there, and hopefully we can take some questions. Doesn't talk too long.
Yes, so then on from question-driven science to just questions about science, I guess. So questions from the audience?
So what was the moment that you changed from chemistry to sort of synthetic biology and genetics? What do you think was did it came by chance? You met someone and you thought oh, this is cool, or you started reading and it was an interest that you picked up, or how did that? Because I think for a scientist it's hard to change from a field to another one, because it's you kind of know that that's your niche, so you go into a new field. So yeah, how was that?
Yeah, that's a great question. What's your name?
Neria.
Neria. Nice to meet you, Neria. So that's a great question. So I hope everyone heard that. She's asking, how did I change from working on being sort of chemistry, biochemistry, structural biology, to doing more of what we're doing now, which is in the area of genetics and synthetic biology. And it was a process, really. So when I was at Yale, I was on the Yale faculty for 8 years. That's where I started my independent career. And we were, very heavily focused on structural biology and biochemistry at that time, and even chemistry. So I had a couple of collaborations with chemistry colleagues. I had somebody doing solid-state NMR on RNA in the lab, and we were trying to apply different sort of chemical methods and biophysical methods to probe RNA structure, which was great. But I began to have this feeling that, because there was a lot happening in genetics and in cell biology in those fields, a lot going on that I wasn't really involved with. And I began to have this feeling that I started to worry a little bit that the work that we were doing on molecules was disconnected to what's happening in biology and in cells in particular, or organisms, where, a lot more complicated. And so I really started to feel like, I wanted our work to be relevant to biology and to make sure that it was. And so I was kind of looking for ways to kind of go in that direction. And then, as chance would have it, I got a job offer at the University of California, Berkeley. And I was really happy at Yale. I wasn't really thinking of moving, but I went and took a look at it. And I realized when I went to Berkeley that, it's a very different kind of place than Yale. It's a public university versus private. It's big versus small. And there was kind of everybody under the sun, was kind of there. You could find somebody working on almost anything. And I thought, gosh, if I move to Berkeley, I will absolutely be able to pursue these connections, and I'll be able to learn the kind of genetics and cell biology that I need to learn to take our research in that direction. And I had no real idea of how I would do it, but it just seemed like it would be very possible to do it there somehow. And so I did make the move, and I started to look for ways to, get more into the field of biology. And the CRISPR project was frankly something that came up very much out of the blue, and it didn't come from a biologist. It actually came from a geologist who, Jill Banfield, who's I guess you could say she's a geobiologist, but she doesn't do any experimental work. She does environmental research, and she was studying genome sequences from bacteria and found these CRISPR systems and wondered if they were an RNA-guided sort of immune system. And anyhow, so I just, it's been a kind of a stepwise process, but it involved, first of all, recognizing that was a direction that I wanted to go, and then looking for ways that I could get the expertise into my lab, either initially through collaboration, but eventually by actually attracting people to my lab who were experienced in those areas. And now in my lab, I have people that are very experienced mouse geneticists. We have animal protocols. We're doing, mouse research, which I could have never imagined doing that before. And we do lots of work with cultured cells, which up until 2012, we really didn't do at all, except for bacterial work. So it's just a kind of an incremental process.
More questions? No, so in that case, I have a question. So you have, during your career, I think, frequently been in the position where you're working with something that nobody has ever done before, right? So for example, with the crystallization of the RNA molecules. So maybe you could comment a little bit on not having that background of a scientific community when getting into a problem and how you approach that.
Okay.
If you understand what I mean.
Yeah. So it's sort of related to your question, I think, in a way. It's sort of how do you go in a direction where there isn't a clear path? Others aren't maybe working in that area right now, and or maybe there are even naysayers, people who say, well, that'll never work, right? And yeah, if I had a nickel for every time somebody told me that about things I wanted to do, I could probably fund a project in the lab. So you have to be willing to kind of trust yourself at some level, and take risk, and decide that you're so passionately interested in trying to do something that you're willing to take that risk. And I think that's what it's often been about for me. It's just that I just get really interested in something, like with the, what I mentioned about the going after RNA structures. I just so desperately wanted to see the structures of these RNAs. They were so cool, they were so fun. We were doing all this biochemistry and they could do amazing things, but what the heck did they look like? How did RNA make an active site? Nobody had a clue at that time, right? And so it just was so tantalizing to think that, this could be possible to do. And I just really, really wanted to do it. And I think that's one element that you have to have. You have to really, really want something, because, what I'm not telling you, but I kind of alluded to, is all of the sleepless nights I had, all the times when things really weren't working, and I really thought, I don't know if I'm ever going to get there, and where you start to think maybe the doubters were right, right? That's and so you have to really, really want to do something. And I've heard this over and over from other scientists too, right? If you ask people, like I was thinking about Brian Kobilka, right? So, the 7-TM receptor, trying to get structures of these very difficult membrane-bound molecules, and, a similar kind of thing. No one had really done it. It was very hard to purify these. Nobody knew even how you would crystallize something like that. But he really, really, really wanted to do it, right? And so he just put in the time to figure it out, step by step by step. And that's sort of what it's always been like for me, for these kinds of projects too. And quite frankly, in a kind of a weird way, I'm happiest usually when I'm going after something like that, right? It's more comfortable when you're going after something where you kind of know that it's going to work out, right? You kind of know. Because you can get very comfortable doing science like that, where you sort of know that, yeah, if I do this experiment, I'm going to get a result, and I'm going to be able to write a paper, and I'm going to be able to get my next grant, and blah, blah, blah. But the flip side is that it's not as exciting, and there's something so energizing about working on something that is really hard and where you absolutely have no idea if it'll pan out at all. But you want to do it because you just think it would be so great if it worked.
My first question is, did you ever feel overwhelmed by your success, and how did you deal with it? And my second question is, have you ever felt that you're treated differently because you're a female leading scientist in the field? And what do you think in general about the situation of leading female scientists in general in science?
Ooh, two great questions. So to the first one about feeling overwhelmed, yes, in different ways. So I don't know how to answer that. I mean, I think that I'll just make a couple of comments. I mean, I think that, first of all, I could have never imagined, where things would end up, and for me, and I don't know where they're going in the future, which is probably a good thing, right? Because I don't know what I'll be working on in a few years, and that's probably a good thing. I think that I always try to stay grounded in the science and the people that I work with. I feel so dedicated to my students. I really, my greatest joys these days come from seeing a student or a former postdoc or somebody, who's come from the lab doing something really exciting, and reading a paper of theirs, or they text me and say, "Hey, Jennifer, I got this great result." And that's really, really fun. So I'm getting to a stage in my career where I'm really thinking about the next generation, right? And what all of you are going to do in the future, which is going to be so important and exciting for science and technology in the future. So I try to focus on that. When I feel overwhelmed, that always keeps me grounded, is thinking about that and what's really important to me, what really matters to me. But yeah, I mean, there's so many balls in the air. There's so much going on. I mean, CRISPR babies, gene drives, the patent fight. There's so many facets to this, and I just try to always view it through a lens of being a student myself. Let's take the patent fight. I knew nothing about patents before all of the CRISPR stuff got going. And so, I view it as an opportunity. It's an opportunity to learn. It's an opportunity for me to learn about something I didn't know about before, meet people I never met before. Most of them are lawyers, But right, I try to always view it that way, as life is an adventure. This is an adventure, and I have to be open to it. And I find that to be a helpful way to think about things. When I start to feel overwhelmed, when I start to feel burdened down by these things, I just say, it's an adventure. We just take the next step and see where it goes. But I'll just tell you one little vignette. So I was invited to a meeting. It was up in the Napa Valley. This was probably, it was probably like 2014, early 2014. So it was, we were a couple years, year and a half or so into the world of CRISPR-Cas9 and people doing genome editing, and you could see the potential of it. And some investing group, some group of investors invited me up to a conference in Napa Valley, and it was at a very fancy place. And so I went to this thing, and I arrived a little bit early in the afternoon. And I don't know if you know the Napa Valley at all, but it's, try to imagine if you know the Mediterranean area, if you've been to Greece, it sort of looks a little bit like that. It's got rolling golden hills, beautiful, California live oaks that are growing, and you can see for a long way, vistas. And so anyway, I arrived a couple of hours early before the conference was starting, so I had a little time to go for a hike. So I took a hike up just a little trail kind of behind this resort up to top of a small hill where I had this kind of vista, and so I hiked up there, and I was just sitting on a rock up there, and I was looking out on this beautiful, beautiful landscape. And I realized that I hadn't really had time to do even that kind of a small hike for a long time at that point, because I'd been running so hard. I'd been running so hard, just trying to write papers and get our science done, and all the stuff that was starting to come at me, with just everything coming at me. I have a son at home, and just trying to balance that. And so I was sitting on the top of this hill, and I was kind of reflecting on that, and I suddenly had this incredible sense of sadness. I felt very, very sad, like deeply sad. And I'm like, why do I feel this tremendous sadness? And I realized that, sort of reflecting on it and thinking about it later, I thought, well, it's really about the before and after of my life. There was my life before CRISPR, when I had time for hiking. I had time to take my son out on the weekends. I had time to, just hang out in my lab and chit-chat about science, and, go out, have a beer with my group occasionally, and all those sorts of things. And I had a garden. I loved gardening, and I had an active vegetable garden that I would work in every weekend. And after CRISPR, that all went away for me, right? That all went away, kind of, because I was doing so much, just to keep all the balls in the air. And so there was kind of this very clear before and after. And I realized that as wonderful as CRISPR had been for me, scientifically, I paid a price for it, and that was kind of, and it's just how it is. And then to your second question about, being a female, a woman in science, I have to say that I feel that for the most part, I've really benefited from great mentors, both men and women, who were very supportive of me at different times in my career, going all the way back to Miss Wong, and so I really never, even in grad school and maybe even during my postdoc, I remember thinking to myself, there's no issue of women in science. What is everybody talking? I mean, I don't see any problem. And it was only later, it was only when I got a bit farther along in my career that I started to, find myself in meetings where I would look around the room and realize, oh, there's, I'm the only woman at this table, that sort of thing. And again, I didn't really feel discrimination, but, I felt like there must be something going on, because I saw 50-50 women in my graduate class, lots of female postdocs that were postdocs when I was, and yet once I got into a faculty position, boy, there weren't very many women on the faculty at Yale, for example, very few. There's a few more at Berkeley, and I think Berkeley's actually done well hiring and tenuring women, but there's still a discrepancy, and there's lots of people have analyzed the reasons for this, but I still think that it is, it's just different being a man versus a woman in science, and it's part of it's biological. If you have children, even though I've got a very, very supportive spouse, which, by the way, I recommend all of you pick your partner wisely. For men and women, this is very important. Pick your partner wisely. If you pick the right partner who understands your passion for science, that's a big leg up, no matter what you do. So fortunately, I have that for sure with Jamie Cate, who's a professor at Berkeley as well, a very accomplished scientist in his own right. But nonetheless, there are just things that mom's got to do, and dad can't really do them for kids, especially when they're very young. And it just it is what it is, and it's not bad. It's just, but you just have to account for it. So I think that I would say today that I guess the one comment I would make about sort of the world that I live in now and my experience in my career right now is that I think that there's a different expectation of women versus men. So I often feel, and let's take Hillary Clinton, for example, okay? Hillary Clinton, who ran for president in 2016 in the US. And regardless of, whether you know her or what you might think about her politically, I think it's pretty clear that she was treated differently because she was a woman compared to a man. And, women, if a woman behaves in an assertive manner, she's often labeled a bitch, right? Or, somehow there's something wrong with her that's not feminine, and so it's not acceptable to behave in a certain way. And so I think that that's something that I notice increasingly that I have to pay attention to myself because of the media attention that I have, right? Is that I need to think about that because people probably, even without even thinking about it, expect a woman to behave in a certain way. And if you step outside of that, immediately people are suspect or critical of it. So
I think we had one more question back here somewhere.
Hi. Just a quick question. So you talked about resilience and how much resilience you actually need to achieve the goals of your project. But sometimes projects just don't fly.
Right.
Just they don't take off. So what's your process when that happens?
Yeah, it's a great question. I mean, it's sort of the question of how do you decide, how do you even decide when a project has failed, right? Sometimes it's not entirely clear. Things might not be working, but you might have the sense that, well, if I do one more experiment, I'm still one experiment away. And believe me, I've been there many times with different things that we've done. And I guess that, so what do I do when a project isn't working? I guess that I've always, for myself, when I was working in the lab and now with people that are working with me, I always encourage them to have more than one thing going at any one time. That's one strategy. So I certainly did that as a grad student. I always would have 2 or 3 things that I was working on. And as you can imagine, they were almost never all working out, but hopefully if things were good, at least one thing was kind of working, so that you always had something that you could look to for, a feeling of, I'm actually getting somewhere with my work. So that's one strategy. And I think it's hard to give you like a formula for, okay, I take this step, and then I decide, oh, that's failed, and it's not really like that for me, at least. It's more of a, kind of a judgment call of saying, I'm always sort of asking how, first of all, it's sort of a risk, cost, benefit kind of thing. You're always asking, how important is this to do versus how hard is it and how risky is it and how much have I already struggled with it, And if something is, and I think maybe I would just also add that I think there's a timeliness to science, don't you think? I mean, I think that, like if I had tried to do the RNA structural work that we tried to do, if I had tried to do that even 5 years earlier, it probably would have failed. And the reason is that Aki Uhlenbeck hadn't yet published the work showing that you could use T7 RNA polymerase to make RNA in vitro. There was a lot of work, the cryocooling hadn't yet really been fully developed. There were all these things, sort of technical things that were enabling that only came online just very shortly before I started doing that work. So I think there's kind of a timeliness to it that matters as well. So I'm always trying to, with projects that we're working on, I'm always trying to figure out, are we too early? If you're too late, you kind of know because somebody's already done it. But if you're too early, sometimes it's hard to tell, right? Am I too early or not? And so I'm always asking that question, and then how important is it to do this? If something is really, really important to do, then you're probably willing to struggle more and take more time to do it, maybe, especially if you have other things that are shorter term projects that you can do kind of in the, in parallel. So those are sort of the things that I go through in my mind. And nowadays, it's not me doing the work in the lab, it's students doing the work. And so I'm also always trying to figure out, I try to figure out the person's personality, right? Is this somebody who, likes that kind of struggle, or is this somebody who gets so frustrated that, it's going to be a career killer for them to have to struggle with this project. It's just not the right fit for them. They need to be doing something different, or working with somebody different. So I'm always trying to do that too, is figure out, do I have the right person on the right project, or is this really just not the right fit? And if it's not the right fit, I try to correct that as quickly as I can. So.
So we have time for one more short question. I think we've had the longest.
Thank you very much for your interesting talk. You mentioned that when you started your more exploratory work, it was very freeing that you didn't have so high expectations onto you from your family or from your surrounding. How do you feel now, where you've had a lot of success? Does that expectations that people have to your research feel more like that it's enhancing more exploratory projects, or that it's more inhibiting, or it doesn't really matter at all?
I think the bar continually goes up, kind of, right? Maybe for all of science, but certainly I try to apply that to myself. Like I remember, when I was at Yale. I'd been at Yale 3 years or so, and I applied, and then I actually got a Howard Hughes Medical Institute professorship, which was great. I mean, it was a lot of money, and it was very enabling for me. I wouldn't have to spend a lot of time writing grants because now I had support for my lab and I could do things. And so I was sitting in Tom Steitz's office at Yale, talking about this, and he had been. I think he was maybe the department chair at the time. Anyway, he had been certainly very helpful to me in putting together the application. And he said, congratulations. And then he immediately said, now, he said to me, the bar has just gone up a lot for you. Right? He said, you have been given something that is very enabling, and now more is expected of you. He said, we expect more of you, and the whole community expects more of you. And you can imagine, there I am, a little assistant professor, like, oh. But he was right, And so I do think that, that the bar continues to go up. And I think for me now, I would say that the way that I view it for myself now, personally, is that the bar is high in terms of my integrity, my scholarship, and my mentorship, right? So it's not for me today now as much about, am I going to, do personally myself do the next big thing? It's, am I going to enable others to do it, right? Enable people in my lab to go off and do that. Enable people that are collaborating with us to do that. Enable people in our institute to have the resources that they need to do that. So that's kind of the bar that I now apply to myself.
Unfortunately not. We've run out of time.
Ask me later.
We all have a tight schedule to stick to. And so once again, thank you very much for this wonderful presentation and for sharing your experiences. And we welcome you all back to the main scientific lecture, which will be given over at the new Biomedicum in the main hall there at 4 o'clock this afternoon. So if we can once again thank Jennifer and
thank you very much.